Welcome everyone to the Jones Seminar today, the special Jones Seminar. I realize we're a little tighter up here in room 200, but looks like there's still lots of empty seats. So thank you for coming. This is the last Jones Seminar of the fall term, and so it's a real pleasure to have Kim Samkoe, who's an assistant professor here in the Department of Surgery at the Geisel School of Medicine and adjunct professor here in engineering science, giving the Jones Seminar today. Just in the way of introduction, she did her Bachelor of Science in Biochemistry with a minor in Mathematics from the University of Regina in Canada. Did a PhD in Biophysical Chemistry in the Department of Chemistry at University of Calgary, Alberta, where she was funded on a National Science and Engineering Research Council Committee grant. She then, in 2007, came as a postdoctoral trainee in biomedical engineering here at Thayer, working with me. And that worked for a couple of years, and that was 10 years ago now. She since joined the Geisel School of Medicine in the Department of Surgery to fund her own lab, and which has now been mapped over to the Dartmouth Hitchcock Health Care System, and so she's a faculty member in Department of Surgery. She was named the Harms Scholar in the Department of Surgery, which is an honor to help young scholars create their own research program. She's been a conference organizer for the SPIE BIOS Clinical Translational and Clinical Applications Conference, for the OSA Optical Probes Imaging and Drug Delivery Conference, for the International Photodynamic Association Conference, and the World Molecular Imaging Congress. She's hosted over 20 undergraduate research students in medical interns and WISP undergrad interns in her lab over these years. She has a large network of both external and internal Dartmouth collaborators with joint NIH funding from a number of grants with Professor Scott Davis, John Elliott, orthopedic surgeon Eric Henderson, senior mentor Tayyaba Hassan at Harvard Med School, as well as Keith Paulson and myself. She's currently funded on an Aspire Award from the Mark Foundation for Cancer Research, as well as some smaller internal grants from the Dartmouth Hitchcock. Most notably, though, she was awarded the NIH R37 Award, which is a special funding category for people who score the highest in their R01 application. And so the top percentile, instead of they ask for 5 years, they give them 7 years of funding. Just to sort of help them kick off their research program. And again, that's merit-based. So her research program involves the clinical translation of molecular targeted fluorescence imaging agents for quantitative detection of tumors and therapeutic monitoring. She has extensive experience in xenograft models of human cancer, good laboratory practice, toxicity and stability testing of imaging agents for human translation, photodynamic therapy, and multiphoton spectroscopy. So Kim.
Great. Thank you. Sorry, I'm getting a message here on my computer. Okay, I'll just leave it. So thank you. Sorry, I thought I had shut all of that stuff down. So thank you for the opportunity to speak today and to tell you a little bit about my research. I've had the pleasure of meeting a lot of you this afternoon or today, and hopefully meet more of you tomorrow. So I just want to make sure that, okay, that's on. Before I get started today, I just want to say that I have some disclosures. I'm going to be talking extensively about this. Of that, sorry, about a drug product called ABY029, which is an exploratory investigational new drug with the FDA. And it was developed in partnership, an academic industrial partnership with both Lycor and Affibody, so there is some industrial contribution. But first I wanted to just tell you a little bit more about where I'm from and what I've done in my academic past. I am from, sorry. I grew up in Canada and obtained my Bachelor of Science from the University of Regina in biochemistry, as Brian mentioned, and moving to biophysical chemistry before moving here, where I've integrated more into the medical imaging. And so really what this has done is allowed me to bridge between understanding small molecules and their interactions with their environment, but actually monitoring it on a microscopic to macroscopic scale. So I started my research career as a protein crystallographer at the University of Regina and doing some small molecule peptide synthesis. But really in my PhD is where I got the interest for biomedical or more biomedical research. I was using biophysical chemistry to study the interactions of photosensitizing agents in multiphoton spectroscopy with the environment. So optical trapping, dipole moment calculations, that sort of thing. And also, but applying this photosensitizing agents to a vascular disease where I demonstrated the first ever closure of single blood vessels using multiphoton photodynamic therapy. But what was really lacking and where my interest still lies is really in imaging biological response. And so I came here to Thayer to work with Brian in biomedical engineering. So really getting a handle on clinical imaging methodologies, small animal oncology models, and quantitative fluorescence imaging. So in my current research track and program, I am highly invested in molecular imaging and looking at biological processes in vivo and actually quantifying processes in vivo that normally we look at ex vivo. So actually trying to translate quantitative imaging to understand and to optimize tumor detection and to understand tumor response to therapies so that we can not only monitor therapeutic response, but we could actually potentially predict therapeutic response as well. And so really what I wanted to talk about today is using these molecular agents, we need to translate them from from the lab to clinic. But in order to do so, you have to understand a broad number of characteristics of how these molecules interact with their environment and how something that acts one way in the lab might not act the same way in the clinic. And so today, I'll be talking about the procedure of taking an agent to clinic and the concepts that need to be considered. So really, today we're going to be talking about integrating two types of cancer therapy. One is surgery, so actually removing tumors from the human body. And if tumors are removed accurately without leaving any cells behind, this can really truly be a curative technique. And then also taking targeted therapeutic agents, which are meant to target biological processes, and using them as imaging agents so that we can look at fluorescence-guided surgery. So as most of you probably know, that optical imaging is both sensitive and specific. And so not only are we able to image with high molecular sensitivity, we can image down to nanomolar or even high picomolar concentrations of fluorescent agents, but we can also image those over several orders of magnitude in terms of resolution, spatial resolution. So up here is an example of a patient that has been administered our ABY029, and we can actually image this in the patient in the clinical setting. We can remove it from the patient and image it on what we call a back table system, where we can look at how close we can use fluorescence to determine margins of the tumor. You can then cross-section the tumor, and you can look at different resolutions, and you can see that at low resolutions, you can still see the tumor location. But at high resolutions, even on thick tissue, that we can really start seeing the cellular information that the molecular targets are giving us. And it's quite comparable to microscopic work where you can actually see whole cells. So, in general, any sort of medical imaging that uses a contrast agent is going to utilize the enhanced permeability and retention effect to garner contrast between the tumor and the normal surrounding tissue. So the enhanced permeability and retention effect is simply that tumors have recruited blood vessels into them to deliver nutrients. These blood vessels are not formed very well. They tend to be leaky, and they leak a lot of interstitial fluid out into the surrounding area. This accumulation of the interstitial fluid plus the uncontrollable growth of the tumor can cause excessive pressures. These pressures can collapse the lymphatic vessel system and actually cause, so we're getting more into the tumor, but we actually are having troubles draining the tumor of these agents. And so what really happens is we get a differential uptake between the tumor and the normal tissue. And this is what's causing most contrast that we use for medical imaging. So, for example, a patient here is injected with an agent. It initially goes to the bloodstream. It's distributed throughout the body. And then the tissue distribution and clearance both play a role in the differential uptake in the tumor, or differential uptake and clearance in the tumor and the normal tissue. So normal tissue tends to clear fairly rapidly, and tumor tissue clears fairly slowly. And so the difference between here is our contrast. And that contrast, the time scale that this happens on, is going to determine what your clinical viable imaging window is going to be. And so really the key point to this is that the distribution and clearance properties of targeted agents can be tailored. So if you know what your molecule is like, you can or the properties of your molecule, you can predict how it's going to act in the body, or at least you can test how it's going to act. So when considering what makes a good clinical agent, obviously long retention in a tumor, fast clearance in the normal tissue, but we're also interested in the target in molecular imaging is highly specific for its target, and there's not a lot of non-specific binding. We also want it to evenly distribute throughout the tissue so that if any of the tissue contains receptors or the actual target for the molecular agent, that it's going to see it. We also don't want to have any toxicities or biological adverse events. And so really when looking from clinical lab to clinical translation, a lot of agents fail really in both clinical efficiency and in human adverse events and animal toxicities. And so these are things that we can potentially predict in preclinical research. When we go from, if you look at the PubMed index articles, fluorescence molecular imaging has several thousand articles, and yet fluorescence guided surgery or fluorescent molecular surgery in clinical trials is very few. And so there's currently a lag between preclinical and clinical research. So the first thing you want to do when you're looking for molecular targeted agents is actually pick a target. What are you interested in the body that you want to be able to visualize? And so one of the most common targets for molecular therapies is actually proteins within the body. So either growth factors, growth factor receptors, protein signaling cascades. These are all indicated in oncology and have a lot of therapeutic agents. You can maybe see some of the therapeutic agents that are listed here. And the nice thing about molecular imaging is you can actually use these targeted agents as your imaging agent as well, as long as you are able to see it. So one of the considerations that you have to think about is, I don't know why those keep coming up, is where your target actually is. Is it in the intracellular space? Is it in the extracellular space? Is it freely diffusing or membrane bound? What are you actually going to be looking at? So for instance, a growth factor receptor is membrane bound, and it actually has two binding sites on the outside of the cell and on the inside of the cell. Intracellular proteins are limited to the cell, but then they're also difficult to get to because you not only have to have plasma delivery, but you need to be able to diffuse through the tumor and into the cell itself. So you have to think about the barriers to delivery. Right. So almost exclusively today, I'm going to be talking about the epidermal growth factor receptor. Is a transmembrane protein receptor that's indicated in the growth migration of cancer. It is a highly sought out enzyme for or receptor for therapeutic agents. As you can see down here in these xenograft models, EGFR is highly overexpressed in different tumor lines, and this is a human cancer model, human cancer where you can see that EGFR is overexpressed in the tumor region compared to the normal. So EGFR is a nice target for oncology because it's not just restricted to one type of cancer. It actually is overexpressed in several cancer types, including head and neck cancer, which is what I'm going to be talking mostly about today. Head and neck cancer, almost every head and neck cancer overexpresses EGFR. So once you have your target, you know where your target is localized, you need to think about what agent you're going to use, how specific it is for the target, and then also the clinical application. And really, these two things are going to go hand in hand. The properties of your agent are going to actually determine their clinical application. So there's a lot of proteins, engineered proteins, and nanoconstructs that are available currently for targeting. Today I'm going to really focus on the two ends. I'm not really going to talk so much about nanoconstructs, but nanoconstructs often have some of these proteins labeled on the outside of it for targeting. But really I'm going to focus on the two ends, an antibody and an affibody. And both of these agents have been developed and put into the clinic, and they both have very different properties which make their usefulness different. So I thought there would be a good comparison. Both of these agents that I'm going to talk about today are labeled with a near-infrared dye called IR dye 800. So the protein production and synthesis vary differently between these two. Antibodies are typically made in animals and then harvested and isolated and then need to be expanded. And in order to expand antibodies, you can do this in cell culture, but you can also do it in an animal model. However, these small molecule affibodies are synthetic peptides that are based on a subunit of the staphylococcal protein, which is known to bind to human immunoglobulins. And so these proteins are actually synthesized with varying amino acid structures and then are tested for specificity. These can then be used in recombinant protein production. However, in clinical applications, recombinant protein production is not necessarily the best because you can have toxic byproducts from the E. coli, which then have to be further purified out of the cell samples or of the product samples. And so another way to produce peptides is through peptide synthesis, which is slightly cleaner in terms of toxic byproducts, but also a lot less inexpensive. The production of a novel antibody costs several $1 million, where the production of a small molecule peptide can be more in the $100,000 range. So once you have a target, so we're talking about antibody and these synthetic peptide affibodies, you need to think about your fluorophore selection and how you're actually going to label those. The wavelength of light that your fluorophore emits and absorbs that is going to determine your depth of imaging and also whether you're going to interfere with biological autofluorescence. And so a lot of the current thrust in fluorescence-guided surgery is to use near-infrared fluorophores, which are outside of this autofluorescence range and have a larger depth of imaging. You also have to think about whether your fluorophore is going to allow your agent to get to its target. So if you have an extracellular receptor, you really only need to get through the plasma system. But if you have an intracellular receptor, you're going to need a fluorophore that can actually get through the cell membrane as well. So the properties of the fluorophores themselves are important. In cyanine dyes, for instance, are popular in near-infrared fluorophores, and they cannot diffuse through the cellular membrane, but rhodamine dyes, for instance, can. So the other portion that can have an effect on your agent is how you actually conjugate your agent to the molecular target. So there are NHS ester labels, which is a fairly nonspecific labeling that binds to primary and secondary lysine groups. Antibodies and proteins in general have a lot of lysine. So for one point, this is very convenient because you know that your antibody or your protein is going to be labeled. It is inconvenient in the fact that it's hard to control the amount of labeling. And you actually end up with a solution of agent that has a different number of labels on it. So even though your average might be 1, you might have some that have 3 labels and some that have none. And why this is important is that the number of labels can actually change how you see it. So if you have too many labels on your protein, you can actually start getting a decrease in fluorescence intensity based on fluorophore interactions. Additionally, where they're bound can actually affect the functionality of your molecular probe. If the fluorophore is bound to a lysine in the binding site, then it might not be able to actually bind to your target and then is not a useful probe. So a lot of synthetic proteins are going the way of using cysteines, which are not a common amino acid, especially in smaller peptide regions. And so this is the way that the affibodies are used. So the cysteine, the proteins are synthesized to have a single cysteine unit on the protein itself. And so we can actually site-specific label it using the maleimide functionalized fluorophores. What this means is that we have a highly homogeneous target population where we have either proteins that don't have fluorophore or proteins that have 1 fluorophore. These are also easier to separate in small molecule peptides because the molecular weight differential is a lot larger. These dyes are about 1 kilodalton, so separating a 1 kilodalton difference in a 6 kilodalton protein versus 152 kilodalton protein is substantial. There's also been some research to show that the site-specific or the number of fluorophores can actually influence where your targeting agent is going to go in the body. So if we look at a nanobody, which is a small peptide, that was labeled in 2 ways. It was labeled non-specifically and then site-specifically. And so the non-specific labeling actually shows this is a mouse laying on its stomach, so you're imaging the back, and then a mouse laying on its back, so we're imaging its abdomen. You can see that we have fluorescence originating from the liver and from the kidneys. However, if we look at site-specific labeling, where we know there's only 1 dye on the agent, we get much more selective clearance in the kidneys. And this becomes important because, as I'm about to go into in some depth, the pharmacokinetics and dynamics of the protein within the body are really what's going to allow for you to determine what imaging time is going to be useful. So the plasma excretion curves of a human or an animal are really going to dictate when and for how long we can image and actually be able to see our target. So these vascular profiles have two phases. The first phase is the distribution phase. And this represents the agent moving from the blood into the tissue, the surrounding tissue. The second phase is the elimination phase. And that's removal from the entire body, and this is mostly done through the liver and renal systems, but it can be done by any excretion, so sweat, tears, all that different work. So the distribution phase here, the faster it drops, the more has actually gotten out into the tissue. So you want to optimize your condition to have a fast initial drop, and then the elimination phase here is really going to determine how long your agent stays around. And so if you take your elimination phase and kind of backtrack it to this, what they call a C0, this is the plasma concentration immediately after injection, if you just injected it into the whole body at one time. But the point of this is that this slope is going to determine the C0. So the flatter the slope, the bigger the difference there is going to be between your injection dose and then this C0. And that really tells you how much of your drug has come out into the surrounding area. And just to indicate this in terms of our little person diagram, that this distribution phase is really coming from vascular to total body distribution. And this is going to rely on the differential clearance between the tumor and the normal tissue. Now, what is key about these facts is these are actually competing events. So what makes something a really good distributor within tissue also makes them clear really quickly from the human body. And so it's an interplay between these two agents is what we're really looking for. So properties that involve tissue distribution. So small molecules are going to have higher tissue distribution than large. Molecules that are positively charged. Protein plasma association, which increases their apparent size, is going to cause low tissue distribution. And if you kind of look back and forth between what makes something clear quickly out of the renal system or slow through the hepatic system and the tissue distribution, you can kind of see that they're opposites of each other. And so it's really an interplay between these two agents, which makes a successful molecular targeted agent. So here's effective molecular weight here on the x-axis and the renal clearance here on the y-axis. And so this is 10 to the 3, so this is about 100 kilodaltons. And so really, the limit for renal clearance is about 60 kilodaltons in size. So anything below 60 is going to preferentially go to the kidneys, and anything above 60 is going to go to the liver. This is also true for hydrodynamic radius. A lot of nanoparticles aren't measured in molecular weight. They're measured in the radius size. And so hydrodynamic radius, the limit for the kidneys is about 5.5. But also to note that this isn't a discrete system. You're not going to just the kidneys or the liver, but it's really a fluctuation between the two. So in order to maximize your tissue delivery and your tissue delivery and clearance, you actually really want to have fairly fast tissue distribution. And then you can see as you swing between kidney and liver, you can actually tailor how long the agent stays within the body. And this is important for thinking about your clinical time windows. So, for example, these are the two agents that I was talking about before. So ABY029, which is a small 8 kilodalton protein, has a fairly rapid tissue decrease and then relatively slow tissue distribution and then relatively slow clearance. However, the half-life for ABY is about 20 minutes in the plasma. If you look at cetuximab, which is a lot larger and is going almost exclusively going to the liver, you see that the tissue distribution is a lot slower, and then also the clearance is slow. And if you notice here that cetuximab is actually being measured in days versus the minutes or hours that Affibody is being measured. And why this is important is that this tissue distribution phase is actually going to tell you when your maximal tumor or tissue intensity is going to be. So you can see here that the end of the tissue distribution phase in ABY029 is approximately 100 minutes, and then the tumor intensity max in the normal muscle tissue is about 100 minutes to 120 minutes. The tumor uptake is a lot slower, but as you can see here that the differential between these two are quite high after about 2 hours. So with ABY, you need to wait at least 2 hours before you start seeing a difference between your tumor and your normal tissue. The cetuximab IRDI, on the other hand, surgery is on day 4, so your distribution is actually a lot longer. You have to inject your patient 2 to 4 days prior to surgery in order to get that same differential. And just on some comparison on some other agents that we've worked with, we have this nanoconstruct over here, which is a theranostic delivery agent that I've been working with in collaboration with Mass General Hospital. And this nanoconstruct is very large. It's about 133 nanometers in diameter. And as you can see, we have maximal distribution in the normal tissue at about 10 to 12 hours. We don't have a plasma curve here, but you can kind of see how you can estimate what the plasma curve would look like. Interestingly, we've also looked at EGF, labeled with IR.800, and EGF is epidermal growth factor. It's the actual natural ligand for EGFR. And its plasma kinetics are similar but slightly different to ABY029, with maximal tissue accumulation in the normal tissue for EGF being an hour. And even though they're relatively similar size, one of the reasons we think this is happening is because it seems like ABY associates with plasma proteins in really high levels, approximately about 80% of what is injected goes to plasma proteins. And this is actually, I think, a good thing, because plasma proteins are actually delivered at high levels to tumors, because tumors tend to have this leaky vasculature. And then it also slows down clearance enough that it's actually beneficial. And so what we've done with ABY is, instead of with the antibody, you need several days to get into the clinic. But ABY allows us to actually inject the agent in the prior to entering the surgical suite, and have enough tissue clearance to image during the surgery itself. So now that we know how the blood plasma kinetics influenced tumor contrast, the real question is, what does molecular targeting actually do? If we can get contrast with untargeted agents, why do we want to have molecular targeted targeting? So as I've said before, we have non-specific uptake. We're still going to get contrast because of this EPR effect. But what the molecular targeting actually does is it extends the window in which you're able to image, because it, the stronger a molecule binds to its target, the longer it's going to stay there. And so what it actually does is it slows down clearance of the tumor even more. So you have a bigger differential between the tumor and the normal tissue. Now, of course, like everything, there's, so with targeting, you can increase contrast, 2 to 5 times compared to 2 to 3 times of a non-targeted agent. So the binding strength and receptor concentration of your tissue actually really increase the tissue retention. And so what happens is, these are 3 examples of molecules. We have cetuximab, we have EGF, and ABY, and their binding strengths differ by about an order of magnitude. And if you look at the receptor ligand kinetics, you can see that it's highly dependent on the on and off rate to the ligand. So what's really important for molecular targeting is a long off rate. You want something to bind, and you want it to stay there for a while. The faster it comes off, the faster it can be cleared. And so the on rate almost doesn't matter as much as the off rate. But as I said before, there is sort of a compromise to everything. And there's what's called a binding site barrier. So agents that have really strong binding affinities actually have lower tissue distribution than agents that have smaller or lower binding affinities. And this might seem counterintuitive at first, but what happens with, I don't know how well you can see that, but what happens with high affinity agents is it comes out of the blood vessel and it binds the first thing it sees, because the affinity for that target is so strong. If it has a high off rate, which means that it's going to stay on that target for a really long time, it's not going to budge. So everything comes out of the plasma, binds immediately, and stays there. Agents that have lower affinities have a tendency to not bind everything they see. They're going to be able to diffuse through the tumor before they find a receptor that they can bind to. And what this looks like is this group here synthesized these nanobodies and synthesized them, changed the amino acid structure to actually affect the binding affinity. So they had 4 orders of magnitude difference between the binding affinities. And so here, when you look at the low dissociation constant, so this means this does not bind very strongly. You can't see it very well, but here's the blood vessel, and I hope you can kind of see that there's red all the way through. Red is the agent we're looking for. On the other hand, the same nanobody, or with very similar structure nanobody, that has a very high binding affinity, comes out of the blood vessel and immediately binds to everything it sees, and it doesn't totally give you tissue distribution. So if we are looking for tumor margins and we want to know where the tumor meets the normal tissue, having something with a very high binding affinity isn't going to evenly distribute through your tissue. And so it's not going to tell you necessarily where everything is located. And so you really want a moderate binding strength. So the optimal range that they found is, in the 10 to 100 nanomolar range as being optimal imaging agents. So in our group, we also looked at compared the diffusion or the tissue distribution of cetuximab to Affibody, both labeled with fluorophores. And as you can, I think, hopefully see, so GFP, this is in mouse glioma xenograft model. The cell line that we injected had a green fluorescent protein transfected into it, so we could actually see the ground truth, where is every tumor cell that's available. You can see here that these two agents were co-injected with different fluorophores, and the distribution throughout the tumor is a little bit different. As you can see, that Affibody distributed more towards the outside of the tumor edges, and the cetuximab was limited to the inside. And so we did actually see that Affibody not only penetrated further into the tissue, but it highlighted the margins a little bit better than the larger, higher binding strength cetuximab. So the other thing that we have to consider is the toxicity of the agent that you're using itself. There's all sorts of toxicities in the human body. You want to think about how it's interacting with different organs and tissues. In particular, the central nervous system is important. You don't want things going to the brain that shouldn't be in the brain. The cardiovascular system is important. And also the sexual organs. You also need to look at acute and long-term toxicities, looking for weight loss, organ function, blood chemistries. These are all ways to monitor, is your agent doing anything in the body that it shouldn't? Is your body, is the animal or organism reacting in any way to the agent? The other thing to consider is metabolites. If it's being brought into tumor cells, is it being metabolized in the tumor cells? If it's hepatic clearance, is the liver breaking it down? So not only is your agent toxic, but are the byproducts of the metabolism toxic as well? And one thing that we have to consider in the imaging world, especially in using fluorescence, is light toxicity. So are we causing any cellular death from the reaction of light with fluorescent agents? And some of you might know that there are a whole line of photosensitizing agents that react to light and cause tissue damage. So for ABY029, we did extensive toxicity testing here at Dartmouth, looking at tissue biodistribution, light toxicity, and then just pages and pages and pages of blood chemistries and hematology, which I won't show you because they're pretty boring. But really what we found is that the Affibody agent has no noticeable toxicity that we could see at even really high doses. And this was not done by our group, but there's a group that compared the tissue metabolism. So this is within a tumor. They're looking at the metabolic byproducts of the Affibody 800, an antibody called panitumumab 800, and then EGF, the ligand. And what they found was that the antibody is internalized into the cell and actually is metabolized and has a lot of byproducts, which are potential methods for toxicity, whereas the Affibody agent did not have any. But one of the things that we actually took advantage of and can be taken advantage in the imaging world is that even though we're using therapeutic agents, you don't have to administer these at therapeutic doses. And so actually you can administer them in really low doses and achieve subtherapeutic effects because they're not at high enough concentrations to cause any sort of pharmacological effect. So, all right, so in kind of going over these properties, I hope that I've fairly convinced you that that the properties of the agent actually determine when and how you can image. And so just to kind of summarize all of this, we have a really large, strongly binding antibody, and we have a smaller, more weakly binding Affibody. The plasma half-life is in a difference of minutes to days, and so the size is also taking it between renal and kidney clearance. And really what that means is that our administration to imaging time, not injection time, for an antibody, like I said earlier, is 2 to 4 days. And the administration to imaging time of the Affibody is more like 3 to 8 hours. And so this really changes the clinical workflow. And there's actually, again, pluses and minuses to both of these. When you're looking at an antibody that was injected several days before, throughout the period of your surgery, the imaging contrast is probably going to be fairly constant. And so it's not so much of a worry if a patient comes in and then is injected with the agent, and then their surgery is delayed by 4 hours. You don't have to really worry about that optimal imaging window because you have maybe even several days for it. In terms of Affibody, we have the bonus that we can inject before the surgery starts on the same day, so we don't have to have patients coming in using clinical time to receive the drug dose, but then we are more limited in the time frame. This is also important when I talk about the clinical trials because we have this in several different clinical trials here at Dartmouth. One is sarcoma, soft tissue sarcomas. Soft tissue sarcomas of the extremities are only a 2 to 4 hour surgery in general. We also have it in head and neck, but the head and neck surgeries can be upwards of 8 to 12 hours because of the complexity. And so you can see even with a single agent, you have to know what is your clinical application and does your agent fit within that realm. And so just to move on, day of surgery. So just to move on to the clinical trial that we're doing here at Dartmouth, we're using ABY029 in what's called a phase 0 clinical trial. A phase 0 is a microdosing study, so actually using, like I previously stated, sub-pharmacological doses in order to see if the agent, if we can see the agent, and does it localize with EGFR like we think it should. I'm going to summarize the whole clinical trial that we've done so far in 2 slides, so just know that this was a lot of work. However, so far within the 3 clinical trials, we've had 32 patients. We have had no adverse events due to the imaging agent itself. We are seeing, as you can see here, we're able to see the ABY029 in large tissue sections and also at higher resolution scanning, and we can see that the fluorescent matches well to pathology. So here is EGFR immunohistochemistry, so the brown color correlates to where the EGFR is, and you can see that that patterning is matched fairly well by the agent. This is the soft tissue sarcoma, and we can see the same thing over here in the head and neck. And really what I want to also kind of highlight is that even on thick tissue sections, not using microscopy, we're using a fluorescent scanning system, we can actually start seeing cellular structure. We can actually see the molecular agent binding to its target. And so we can truly identify where at least the EGFR expressing tumor is. And you can see here, this is actually native EGF in the mucosa. So EGF is present anyway in the mucosa of the oral cavity. And then you can see the tumor lies over here, and this is non-EGFR expressing cells, of normal tissue. So you can see that we're getting really nice contrast. So in addition, in both of these clinical trials, as we've increased dose, we've seen an increase in our measured fluorescence intensity, which is expected. Interestingly, though our image contrast, we're not fully finished, but we only have 1 or 2 patients left in each trial. We don't actually see a huge advantage in going up to the 6x dose. The 3x dose seems to be giving about the same image contrast. What we do see, though, is a reduction in patient variability. So in the 3x dose, it seems like patients are spread out pretty wide in their uptake of the agent and their clearance, and that seems to stabilize a little bit. The head and neck is an interesting case because, as you saw in the previous image, the normal tissue also expresses EGFR. So although we're getting really nice images, our measured contrast isn't as great because we have normal EGFR expression in the tissue. The other thing I want to just focus on really quickly is that our results correlate very nicely to pathology. So we have the calibrated fluorescence intensity versus the EGFR immunohistochemistry staining intensity. And so we know that not only are we labeling the tumor, but it is highly specific for EGFR. Okay, I'm going to quickly just move on to what we're doing currently and then future, and I think I have a couple of minutes left. So one of the things that we've noticed, a couple of the things that we've noticed in the clinical trial is that in some of the patients, there's really high heterogeneity in the tumors. So EGFR is not labeling specific, or is not labeling all the tissue. It's labeling the EGFR positive tissue, but not necessarily all of the tissue is EGFR positive. And this is especially so in the sarcoma cases. In addition, we also have, as I mentioned previously, different administration to imaging times between the two tumors. So we're also interested in seeing if there's some way that we can optimize that. So in the soft tissue sarcoma study, this is an example, a schematic drawing of what H&E tissue slice would be. So purple in an H&E tissue slice represents viable growing tumors, and pink is staining for proteins. Sorry, purple stains for DNA and pink stains for proteins. And so in general, within a tumor, purple means viable tissue and pink means necrosis or regions that are acellular. So we started to notice that when we imaged with ABY029, which is targeted to EGFR, we had really high correlation between the viable tumor regions and our fluorescence, which makes sense because viable tumor should equal EGFR expression. However, when we use a nonspecific perfusion agent in indocyanine green, we actually saw almost the exact opposite. We saw that indocyanine green, which is not targeted to anything, started pooling in the necrotic regions. And in fact, if you look at a cross-section and correlate the viable tumor to the fluorescence, we actually have a very strong negative correlation, which means the ICG is actually sitting outside of the viable tumor regions. So what we thought is, what if we administer both of these agents, not simultaneously because they have very different uptakes, but what if we administer both of them? Can we actually try to optimize or at least even out the distribution within the tumor? And the answer is yes. It seems like we can. In vivo, we injected ICG 24 hours prior and ABY 4 hours prior to imaging. We excised the tumors, and again, we can look at this tissue distribution, and we see that our Spearman correlation is not zero, so it's not perfectly halfway in between, but it's substantially not correlated, which is something I never thought I'd say. I want to look for no correlation. But what this is really saying is that we're getting even fluorescence from all areas. And so this is actually, we think, a good thing, especially in these soft tissue sarcomas where the margins aren't always getting labeled by our ABY agent. And then just really quickly, it also increased we had the characteristics of the positive characteristics of both ICG and ABY in each case. We had high fluorescence intensity, but we also had high image contrast values, which is really promising. And so really where we're working with this is moving forward is trying to figure out ways that we can label not only tumors that are EGFR specific, but might have other targeting entities, necrotic areas, but also potentially the peritumoral region. We can use activated enzymes like ProSense, which look at tumor microenvironment instead of cell-specific. So there's possibilities that we can actually use multiple agents to optimize tissue contrast. And then just quickly, I have like 2 more slides. Another way that we're looking at using multiple agents to optimize tumor detection is using what we call paired agent imaging. And what paired agent imaging is doing is we're injecting 2 agents that their plasma curves and tissue distributions are matched, so they're identical. And what we do, only one of those agents can bind to the receptor of choice. And so what this actually does is we use the untargeted agent as basically a control to remove all the nonspecific contribution from the targeting agent. And so what that leaves us with is actually the molecules that are actually bound to the targeting agent themselves. Everything else gets normalized out. And so this binding potential is actually proportional to both the target and ligand binding affinity and the available receptor concentration. And so really what we can do is that at time points past injection, we can actually just do an image ratio to get this binding potential information out. In the past, moving from my postdoctoral fellow into my own research program, we showed that paired agent imaging is highly specific, so that it highly accurately reports on receptor concentration. It's also highly sensitive. In this lymph node study, we could detect down to 200 cancer cells. So where we've been moving from here is actually demonstrating that paired agent imaging has both high spatial and temporal diagnostic accuracy. And what that means is, because we're removing the nonspecific binding, we can start imaging almost immediately after injection, because the two agents are going everywhere exactly the same manner. And so we've actually shown that in as little as 30 minutes post-injection, we can start imaging with fairly high diagnostic accuracy. And so this means, in terms of the two surgeries that I told you about, sarcoma, which is fairly short, and head and neck is really long, you could potentially inject in the surgical suite and wait 30 minutes prior to excising the tumor, instead of injecting in the preoperative room. So it gives you some more flexibility in your optimal imaging time. In addition, because fluorescence-guided surgery is really imaging intensity, there's high variability between patients, as I showed you in our clinical trial. And we also showed that in mice with tumors of different EGFR expression, that when we group all of the cohorts together, that fluorescence intensity alone does not have as high of diagnostic accuracy as this paired agent imaging. And that's really because we're imaging receptor concentration, which is independent of patient dose of the receptors or the imaging agent that they received. And this is my final slide, just showing that we're looking at different imaging agents that can be used for paired agent imaging in the clinic. IR-700 is being used in some photoimmunotherapy studies, so it's already in clinical trials. Methylene blue is already clinically approved for use. And this IR-680 is not, but there's interest in actually bringing it to the clinic. And so you can see that in normal tissue, where there's devoid of receptor, these two agents should act exactly the same way. All the studies that I showed you before were done with this IR-700, and you can see that they're not perfectly matched. And so this might be the discrepancy of why we have to wait even 30 minutes. But if you look down the line at IR-7 or 680, they're actually really well matched and potentially might be an even better paired agent for clinical translation. The other bonus is that IR-680 has a really high quantum efficiency, and so you actually can inject less to get the same amount of fluorescence, so you're reducing the potential for toxicity. So I hope that I've conveyed to you today the importance of knowing your molecular imaging agent, how the properties of that imaging agent can affect your clinical outcome and your clinical use. And then I'd like to thank the funding agencies. I'm personally funded, as mentioned before, on an R37 National Cancer Institute award, an Aspire award, and a faculty award through the Department of Surgery. Then also the clinical trial has been funded through an R01 with Keith Paulson as the principal investigator. And then, of course, I'd like to thank the members of my research group, and of course, all of my collaborators here at Dartmouth and nationally. So thank you for your time.